Kocot.
Mm-hmm. Mm-hmm.
All right, I think we're about ready to start. So it is my pleasure today to introduce Kevin Kocot. Sorry, I have a cold. He's from University of Alabama, where he's an assistant professor there, and also the curator of invertebrates at the Alabama Museum of Natural History. He's been there since 2016. And before that, he did his PhD at Auburn with Ken Halanich. Some of you may know Ken. And he did a postdoc at Auburn and then also at the University of Queensland in Australia. So Kevin's going to be talking to us today about biodiversity systematics and genomics of the eculiferan mollusks.
All right. Thanks a lot, Kelly. Thanks for that nice invitation. And thanks for that amazing parade yesterday. Man, you all really know how to welcome a seminar speaker. That's as good as the jokes are going to get. I apologize in advance. No, but seriously, thank you so much for inviting me to come down. It's really nice to be here. There's not so many marine folks in Tuscaloosa, so it's really nice to be here and be interacting with you all. I enjoyed some nice chats this morning. Looking forward to chatting with more of you this afternoon. So today I'm going to be talking about work that my lab has been doing at UA Tuscaloosa over the last 3 years. And it was really fun for me to put this talk together because, frankly, I've given a couple other invited seminars over the last couple of years, and they're mostly kind of cannibalized from my job talk. But my lab's kind of the point now where we have a number of projects that are starting to come to fruition, and a lot of data that's coming out for my graduate students and postdoc. And so I'll be sharing about some of the work we've been doing, looking at a somewhat obscure group of mollusks that you all might not know a lot about, but I think are really evolutionarily important. And so I always like to begin talks telling you that I'm a systematist, so my lab studies taxonomy and systematics. We're interested in describing new species and understanding the evolutionary relationships among different groups of organisms. And the point I want to make is that taxonomy and systematics aren't stamp collecting. It's not got to catch them all, but by knowing the evolutionary relationships among different groups of organisms, we produce a phylogenetic framework that is useful for all aspects of comparative biology. And so this is developing a tool that folks can use to answer any sort of comparative or integrative questions. And so I often joke that we're a bit taxonomically promiscuous. My lab works on everything from tunicates to ctenophores to nematodes to chinorynchs, and we keep picking up more bad habits of invertebrates. I like anything without a spine, basically. But the group that we primarily study are the mollusks, so snails, slugs, clams, squid, octopus, etc. I reckon that many of these animals are very familiar to most of you. But in particular, some that might not be as familiar are the chitons or polyplacophorans, which I'll be talking about a bit today. And then particularly obscure among the mollusks are these two groups, the selenogasters and cautofoveates, which are collectively called the aplacophoran mollusk, which is Latin for without a shell. And so there's two kind of flavors of aplacophorans. The selenogasters have a narrow mid-ventral foot, so they're kind of slug-like. And the cautofoveates completely lack a foot, and they're really honorary worms. They've completely lost the foot and burrow in mud. So I'll be talking about these two groups quite a bit today. So mollusks are, in addition to being diverse, there's about 100,000 described extant species in 8 morphologically really disparate major clades. We have everything from microscopic worms that live between grains of sand to the colossal squid. But they're also important to humans for a number of reasons. So there's lots of economically important species, as I know many of you know. In terms of food, producers of pearls and shells. Am I still on? Oh, there we go. Okay. So a lot of mollusks that are negatively economically important, causing harm. Many mollusks are hosts of human parasites, like the snail biome Filaria. A lot of mollusks, like the sea heraplegia, are model systems for studying learning and memory. Here in Alabama, of course, many freshwater mollusks are of great conservation concern, particularly the freshwater bivalves in the family Unionidae. And to my geological colleagues, many mollusks are useful markers of geological strata. So despite their diversity and importance, the evolutionary relationships among mollusks have long been debated. And so I'm briefly going to talk about some of the work I did during my PhD to try to address this problem. And basically there's been 2 major schools of thought, and these have largely centered around those worm-like aplacophoran mollusks and the chitons. So we know that mollusks are related to annelids, segmented worms. They have very similar larval types. And so some people thought, well, mollusks and annelids must be closely related, and there's worm-like mollusks. So the worm-like mollusks are probably basal and ancestral and primitive, if you will, and thought that these 2 groups of worm-like mollusks made up some sort of like basal paraphyletic grade, sister to all other mollusks. Whereas other folks have noted that the chitons, which have both shells and calcareous scales or sclerites that cover their body, as do the two groups of aplacophorans, probably form a clade, and that the aplacophorans are kind of secondarily pedomorphic or simplified animals. And you're probably wondering, like, well, who cares? But the point is that these two different phylogenetic scenarios tell us very different things about the origin and evolutionary history of this really diverse and interesting group. So if these worm-like aplacophorans are this kind of basal grade, then we can infer that the last common ancestor of mollusca would have been a worm-like animal. Now, on the other hand, if the aplacophorans are secondarily, simplified, or they've taken on this worm-like body plan over evolutionary time from maybe a more chiton-like ancestor, that tells us that the last common ancestor of mollusks was a more slug-like, chiton-like, monoplacophoran-like sort of animal. And so understanding between these two different hypotheses could tell us a lot about the early evolution of this diverse and important group. The problem is there's been a lot of debate in the literature, and different studies have come to really different conclusions. And so this is a review from 2015 by Julia Sigwart and David Lindberg, where they did a meta-analysis of all the different trees that have been published about molluscan phylogeny. And if you take a consensus of all these trees, you get a total polytomy. Every possible scenario has been proposed. And that's in part because we're looking at the morphology and comparing things like colossal squid and microscopic worms that live between grains and sand. It's hard to know if we're comparing apples to apples in terms of morphological characters. Then in terms of molecular data, most molecular studies to date on mollusks have used kind of the usual suspect sort of markers, so 18S, 28S, 16S, CO1. And we see that in different groups of mollusks, there's dramatically different rates of sequence evolution. So for example, if you build an 18S tree with cephalopods, their branch lengths, it's like they're from the future. They're so long branched. And so these different trees have given really dramatically different results. And so for my PhD work, I wanted to use a so-called phylogenomic approach. And so back in 2013, when I was doing this work, 454 pyrosequence, and more like 2011, 454 pyrosequencing was kind of all the rage, right? And so we could get transcriptome data cheaply and easily. And so folks were using these sort of high-throughput sequencing approaches and a phylogenomic approach to study evolutionary relationships using lots of nuclear protein-coding genes derived from genomes or transcriptomes. And around that time, a number of studies came out applying these sort of approaches to a number of different groups whose phylogeny had been recalcitrant to the sort of usual suspect markers. And so in phylogenomics, we don't do PCR to pick out particular genes of interest. We shotgun sequence, the transcriptome or the genome, and pull out particular genes that meet certain criteria. So for example, we need genes that are sampled for most taxa, so we have fairly complete data matrices. We also need sequences that are appropriately conserved for the question of interest. And so I was looking at mollusk class level phylogeny, so we need fairly conserved genes that aren't evolving too rapidly because we have this ancient divergence that probably happened in the early Cambrian. And then finally, we need genes that are orthologous, that is, related by speciation and not gene duplication. This can be kind of challenging in phylogenomics where we just have transcriptomes, maybe a snapshot of genes turned on in a tissue at a given time, but not the entire genome. And so long story short, this is part of the work I did during my PhD dissertation. And as you can see, we have this clade of the two groups of worm-like mollusks as the sister group to the chitons, forming this clade called Aculifera. Consistent with morphological-based hypotheses that suggested that the aplacophorans are probably secondarily vermiform. They probably evolved from a chiton-like ancestor. And so since this work came out, another study actually the same year also had support for this Aculifera and Conchifera dichotomy, or the aplacophoran chiton clade and the clade of all other shelled mollusks. We disagreed on some minor details. For example, the scaphopods were ambiguously placed in both studies. And I also thought I'd mention that this group recovered the monoplacopherans, a group who were unable to sample these deep-sea limpet-like mollusks that were traditionally thought to be kind of the sister group to all other shelled mollusks, as they're kind of simple in morphology and have serially repeated structures, actually with cephalopods. So you have these tiny limpet-like, snail-like animals with cephalopods, which is kind of strange because, of course, cephalopods are highly advanced and derived mollusks. But we know cephalopods are highly derived and advanced, and so they may well have evolved from a simple limpet-like ancestor. And so this is something we've been looking more into. We recently got our hands on some more monoplacophoran material and sequenced a draft genome from the Antarctic monoplacophoran Levopolina antarctica. And so long story short, if we use a maximum likelihood approach with sort of standard approaches using site homogeneous models, we recover the same results of the monoplacopherans and cephalopods with kind of moderate support. But when we use fancier models, basically these so-called site heterogeneous models that better account for site-specific rate heterogeneity. So these problems I was talking about where like cephalopods seem to evolve quickly and they have long branches and molecular phylogenies. We use these sort of models, we get the more traditional approach with the monoplacophoran sister to all other shelled mollusks with fairly strong support. And so it seems that using highly, I should say best fitting models is really important for these sort of issues. And so anyway, My lab's continuing to work on some of this deep molluscan phylogeny work. I have a really talented undergraduate in my lab who's been using so-called ultra-conserved elements to use another source of molecular data to try to tackle this question and confirm this aculifer-conchifer dichotomy, which she's consistently supporting, and address the placement of these monoplacophorans. And it seems to bounce around a bit depending on what source of data we use, where these monoplacophorans fit in. So there's something interesting going on there. But regardless, we're consistently getting this group, the aculiferan mollusks, chitons and aplacophorans, is a monophyletic group, sister to all the other mollusks. So this is a group I've gotten really interested in because there are many people worldwide studying things like gastropods and bivalves, and of course these are really diverse and interesting and important groups of mollusks. But in terms of these worm-like aplacophoran mollusks and chitons, there's just a handful of people worldwide actively working on them right now. And basically everywhere I go and find nice coarse sand and go digging, I find new species of aplacopherans. They're very abundant, and there's just a handful of people studying them. And so briefly, I think folks might be familiar with chitons already. Again, these are slug-like animals with plates on their back. They have multiple shells, but they also have calcareous sclerites or scales kind of lateral to those shells in this girdle. And then the aplacophorans are a group that I reckon many people aren't super familiar with. It's a really morphologically heterogeneous and diverse group of worm-like animals. There's two general flavors, the caudofoveates, which are burrowing. Again, they've secondarily lost the foot. They have this kind of weird structure around their mouth called the oral shield, which might actually be a vestige of the foot that they use in burrowing. The other group, the selenogasters, are epibenthic or sometimes burrowing. A lot of them wrap around cnidarians. And so these primarily feed on corals or hydroids, whereas the caudofoveates burrow, and we don't really know what they eat, maybe foraminiferans. We've seen nematode contamination in some molecular data. And so this is a group that I've become really fond of because of they're just kind of understudied and underloved, and there's a lot of diversity that's not yet really well known. And so they're mostly deep sea, and that's in part why I think a lot of people aren't working on them. But I've been fortunate to get to tag along on some research expeditions, for example, on the ship Meteor. So in 2011, I was on a Circum Icelandic cruise with this German group. I don't speak any German, so I don't know what the hell was going on most of the time, but I got lots of really wonderful samples. We were doing constant epibenthic sled sampling. I've also been really fortunate to go to the Antarctic a few times and sample on the Gould and the Palmer and also the German ship Polarstern. And this group seems to be particularly diverse in the Antarctic, which is really interesting. So here's me on a particularly bad hair day in Antarctica where I'm sieving some of the mud we dredged up, and then I'll take this under my microscope, and as the samples are doing this under the scope, try to pick out all these small aplacophoran mollusks. But I also work in some warm places sometimes, so I did a postdoc at the University of Queensland in Australia and worked here on Heron Island. So this is a really wonderful place to work. They have really state-of-the-art laboratory facilities. You can get up in the morning and go diving right off the reef, go snorkeling, grab samples. And then at 5, you can walk over to the other side of the island, which is a resort, and have a gin and tonic. So it's a really great place to work. And so for my postdoc, what I wanted to do is get samples of this particular animal. This is Epimenia australis. It's a giant selenogaster. They're about 20 centimeters long, whereas most are maybe a centimeter at most. I flipped over every rock on Heron Island, I think, and I found one. So that didn't go exactly as planned. But this is a really large-bodied animal whose development is well known. And so something my lab is hoping to do in the future is get these animals in the lab and do some developmental work to study developmental gene expression associated with the sclerites and compare those to the shells of other mollusks, which I'll talk about more later. Just some more pretty pictures of this group. So this is another aplacophoran. This is a selenogaster from Antarctica crawling on the surface tension of the water. And you can see they are proper mollusks. They do have a foot. But it also looks kind of shiny because of those sclerites. Again, those are fairly big animals. Here's some more typical ones in terms of body size. Many of them are very small. Some are myofaunal. Others are on the order of a centimeter to just a couple millimeters. Aplacopherans are covered with these calcareous scales, which I mentioned. And so in my lab, we have a tabletop scanning electron microscope, which we use the heck out of. And it's super useful for seeing the differences among these different types of sclerotomes, the different sclerites and scales covering their body. And so this is really useful for identifying specimens. Some of these animals are really beautiful. So this is an undescribed species from the Caribbean that I only know of from photos that divers posted to Facebook. Here's another one that's been all over Facebook, so it's really well known, but undescribed as of yet. We're currently working on describing this one and have settled on the name Dunderzia gaga for, I think, obvious reasons. The strangest place I've ever found an aplacophoran was for sale on a shell collector's website. So this is a woman in Uruguay who was working with a fisherman, and she was buying all the weird stuff that came up in his trawls. So I was able to get my hands on some specimens that unconventional way. So in my lab, the workflow that we're primarily using is to get samples from these large-scale deep-sea expeditions, for example, the Ice Age cruise, where lots of epibenthic sled trawls are being done, they're being bulk fixed, and then sorted in the lab. And so I have a team of awesome students, like my undergrad Carter here, who's imaging every single individual specimen using a stereo microscope that's motorized and connected to the computer. So we can take multiple images at different focal planes, stitch them together, and have a nice reference image. And then we'll do a lot of DNA barcoding as well. And so for those specimens, we'll first do scanning electron microscopy and our little tabletop SEM. Because it's a low accelerating voltage, it doesn't really fry the specimen, so the DNA isn't really degraded that much. And so we can still extract high-quality DNA for barcoding. We're also doing a lot of histology. This is my master's student, Maddie, who's doing histology. And unfortunately, a lot of apelcopherans look really similar, even if you do SEM. And so we have to do histology to look at some internal structures like the radula or the teeth and other reproductive structures to identify them. So here's some slides from my postdoc, Carmen, who just joined my lab about a month ago. So Carmen's been working on some material from the Paris Museum from a few areas in the world where no one has ever really looked at the apelcopheran fauna. And surprise, surprise, she's found some new species. And so she's working to classify and sort these animals into morphospecies, that is, bins of you're different from you, you're different from you. She's trying to identify them, and for that she's having to use classical histology and also scanning electron microscopy. And one of the big goals of my lab right now is to provide then a DNA barcode library so that non-experts in the future will be able to identify animals without the tedious additional labor of doing histology. And so we can take a piece of an animal and do barcoding rather than do histology or something like that. And so here's just some of the animals that Carmen has found in material from the Paris Museum. She's been working in the China Sea and the Caribbean Sea. And so in the China Sea, she's found one species of this genus Undersia. And so she's been doing sorting, looking at their sclerites, those calcareous structures under the SEM and the light microscope. She's been doing DNA barcoding and histology. And in the Caribbean Sea, a lot more diversity there, and she's got a lot of new species that she's working on describing, which is really exciting. We've also been doing a lot of barcoding to look at how many species are there in an area. And a lot of this started where I had an undergraduate who wanted to work in my lab and do a project. And I said, oh, we've got this one species from all around Iceland, haha. So why don't you do barcoding? We'll do population genetics. And instead what she's found is we've got many distinct lineages that are clearly multiple species based on molecular data. And the cool thing is we've done SEM on every single specimen before we did the DNA extraction. So we could go back and look at our SEMs and say, well, sure enough, each clade has these very clade-specific sclerite differences that are subtle and hard to detect, but we're able to see morphological differences as well. So now we're going back and bringing in histology to help clarify the morphological differences among these different groups and formally name some of these species. This is just another species we recently described, and this is a really strange animal from northeastern Iceland, where we collected them in the lab and saw these coming out of them. So here's the head end of the animal, here's the tail end. And this is a strange species that actually broods. Most apelcopherans lay their eggs and crawl away, but this one actually broods its babies in its butt. So that's the mantle cavity there, it's basically the rectum. And we looked, and in the mantle cavity, it's packed full of these little juveniles, which we saw in histology. And so unlike most apelcopheran mollusks, which have a free-living pelagic larva, they actually directly develop inside the egg, and we have a little juvenile inside the adult. So kind of a strange and unknown previously biotic interaction. Another really bizarre species that we've been working with recently is this thing from Antarctica. When we were trawling in Antarctica, we brought up some giant glass sponges, some hexactinellids, and started opening them up to look for worms and things inside. And we found this thing. And I felt really inadequate as a taxonomist because I couldn't even put it into a major clade of mollusks. I'm like, well, it looks kind of like a selenogaster because it's got this red blood. It doesn't have a foot. So it can't be a selenogaster, though, but it doesn't have an oral shield, so it can't be a caudofoviate. And so we started doing histology back in the lab. And if you remember your basic zoology, kind of the defining characters of mollusk is that it has a foot, a radula, and a mantle cavity. Well, this thing has no radula, just kind of this muscular pumping pharynx. It has no foot, it has cuticle all the way around its body. These holes are where the sclerites, those calcareous spines used to be before they were decalcified away. And it has no mantle cavity. At the tail end of the body, the anus just kind of ends. And so we have this highly derived, almost like parasitic sort of morphology of a mollusk, and to really know what to make of it. And so what we've been wanting to do is build a phylogenetic framework for the apelcopherans and include this thing in it to try to figure out how we found the new class of mollusk, and with visions of nature papers dancing in my hair, or head rather. And so to do this, we use a so-called phylogenomic approach again. So briefly, we extract RNA, we make complementary DNA libraries, we turn RNA to cDNA, we sequence it using the Illumina platform, we assemble those transcriptomes, we identify orthologous sequences among the different transcriptomes, we concatenate supermatrices and build trees. And so this is the tree we recovered. And so there's two major groups of apelcopherans. Again, the selenogasters, that's the ones with the foot, and the caudofoviates, the ones that have lost the foot and burrow. And lo and behold, here's our bizarre thing, which we're calling Apotemenia enigmatica. And so this is a selenogaster, a group where all members that have been described so far have a radula, a foot, and a proper mantle cavity. And this bizarre sponge-dwelling animal has completely lost all these structures, which we don't know what to make of that other than it's some sort of parasite-like syndrome where it's secondarily lost a lot of morphological complexity. Notably, there are terabellid polychaetes that live inside the sponge, and there are some other aplacophorans that have seemed to have host switched from corals to or other polychaetes, so maybe it's eating those. We don't know. So I won't go into a whole lot of detail about this phylogeny, but suffice it to say the traditional taxonomy of this group, which is based primarily on the structure of those sclerites, those calcareous scales or spines covering the body, has not really held up. And so, for example, this group in pink here, this group called Cavabolonia, which means that they have a cavity bears, a hollow cavity in their sclerites. It's not a monophyletic grouping. And so it seems that either hollow cavities evolved multiple times, or that was the ancestral condition that was lost multiple times. We've also been using mitochondrial genomes, in part because we can't get RNA from a lot of these samples. I have a lot of material that was bulk fixed on cruises or from museums and where we can only get DNA and it might be degraded. And so working with my collaborator Nina Mikkelsen from Norway, who's currently visiting my lab, we've been working with the Cotophoviates, the group without a foot, to try to sequence mitochondrial genomes and infer the phylogenetic relationships among these different groups. And one thing that's really jumped out at us is the traditional taxonomy of this group, this family called Limophosauridae, which is thought to be sort of primitive, if you will, in their morphology. It turns out to be a non-monophyletic grouping. And we just got our hands on a representative of another genus from this family that we haven't yet sequenced. And so my master's student, Maddie, is going to be adding this other putatively plesiomorphic aplicophoran to this mix. But it seems that, again, in this group, the traditional taxonomy is in need of revision. These families are not monophyletic. This is some other work that Nina did for part of her PhD dissertation. And so she did a higher level phylogeny for the Cotophoviates. Again, that's the group without the foot. And at the genus level, the taxonomy of this group is just a mess. So for example, this genus Spathoderma, it's just peppered all over this subtree. And likewise, there's this genus Falcidens, whereas Falcidens and Chaetoderma are totally intermixed. And so the characters that have been used in the past for classifying these groups are not the characters we should be using, basically. And so this kind of leads us to this state of knowledge where we know that the taxonomy needs some revision. We know that there's a lot of unknown species. And so some of the things that I'm interested in tackling are basic questions like how many species are out there? How many things that we think they're one species, like the species from Iceland, are actually many species? What are the evolutionary relationships among major chitin and aplicophoran lineages? And so the tree I showed you based on the transcriptomes is really just a start at that. There's like 300 species in this group, and we've sampled maybe 20. So we have a long way to go to figure out the higher level relationships of that group. And finally, how can we reduce the need for like laborious lab techniques to identify these animals? So histology is great for looking at internal anatomy, but it's a lot of work. Then we destroy the sample. And so some of the new directions in my lab, and what do we do when we find just one of something, which kind of goes hand in hand with this non-destructive techniques are needed. And so some of the directions that we're moving in my lab is this new workflow where we do a lot of light microscopy using extended focal imaging to take lots of photos of these sort of three-dimensional animals in different planes and stitch them together to get nice kind of reference photos before we have to destroy maybe a tiny specimen for molecular work. And we're now doing a lot of micro-CT scanning. So microcomputed tomography allows us to image internal anatomy of these specimens in a non-destructive manner. We basically take an X-ray at lots of different rotational angles and then stitch them together to make a 3D model. So we can see internal anatomy without actually destroying the specimens. We can then do SEM if we want, and extract DNA, and do this all from the same specimen. So we're getting about as much information as we possibly can from a single animal. And for a group that's largely deep sea, and often we have just one of them, this is really great because it still allows us to do a lot of different work with one individual. And so here's an example of a microcomputed tomograph from an Antarctic aplicophoran called Entonomenia. And I can show you a video of what some of our micro-CT data look like. And so it's a little bright in here, but hopefully you can see that we can see all of the internal anatomy of this animal, and we can rotate them and cut through them at any different plane we want to. And so this allows us to see internal anatomy and do things like count the number of radular teeth or pick your favorite taxonomic character and obtain that information from these animals, and then put them back in the tube in the museum shelf when we're done without destructively sampling them. We've been doing this with a new instrument, the Zeiss X-Radia Versa system, which is kind of a pseudo-synchrotron system. And so we're able to get these really beautiful images in great detail and high resolution of both large and small animals. And so even tiny myofaunal animals, we're able to see enough to identify them down to the genus level. And so moving forward, I'd like to build on this transcriptome-based phylogeny for the aplicophoran mollusk to try to produce a better, more broadly sampled phylogenetic framework for this group. The problem I mentioned is that for many of these things, we only have material that's been fixed in ethanol, or it's from a museum sample. We don't have fresh live material or material that was in RNA later, so we can't do transcriptomes or whole genomes with long reads. And so we want to do this target enrichment approach where we can enrich DNA libraries for just particular genes of interest. And so this is nice because we don't need RNA, we don't need high molecular weight DNA. And the cool thing about this is you design these probes for particular genes that you've identified to be good molecular markers for phylogenetics, but these probes aren't perfect, and they often catch other stuff like mitochondrial genes that tend to be highly abundant. And so we pull out some of your traditional markers like mitochondrial genes and multi-copy genes like 18S to sort of bycatch. So these are backwards compatible with earlier studies as well. Now the problem with this is we need a reference genome in order to map these, in order to design the probes and map our reads back to them. And so my student Megan, who's interested in the sclerites of apelcophorans and biomineralization, is working to sequence the genome of this handsome worm here. This is a giant Antarctic apelcophoran that's about the size of a mini bagel. Most of them are small, but this is a nice big fat one, so we can get plenty of DNA. And so she's using a hybrid assembly approach with Illumina and Oxford nanopore. And so far, we've just done one Illumina run and one nanopore run. And she already has a fairly decent N50 here. So this nanopore approach is allowing us to generate some pretty high quality hybrid genome assemblies. And so she's planning on doing a few more ion runs to get more nanopore data. We're also doing optical mapping to help improve the contiguity of these genomes. And so by having that, that will allow us to then design these probes for target capture, and will also enable her to look at the genes involved in the sclerite biomineralization pathways. And so this is a blob plot here showing us our data so far. These animals have symbiotic bacteria, it seems, living on them, and maybe even in their hemolymph. And they also feed on cnidarians. And although we use blood from this thing, what you can see here is a lot of these sequences down here that differ in coverage from most of the other sequences are actually from cnidarians or bacteria that seem to have been in their hemolymph. And so we've got some decontamination to do. But as we improve our assembly, it'll be easy to sort those scaffolds from the apelcophoran mollusk ones. We're also working in the other plecopherans, the chitons. So this is my student Rebecca Varney, who's really interested in the radula of chitons. So chitons have these incredible teeth that are hardened by magnetite. And so they're basically as hard and as abrasion resistant as tank armor. And it makes a lot of sense if you're a mollusk chewing on rocks all day trying to extract algae from them. Chitons are also really cool because in some of them they have these calcareous sclerites and scales. And this particular species that we're working on, Acanthoplura granulata, in their shells they have these shell eyes. So these sensory pores or aesthetes are modified into image-forming eyes with a mineralized lens. They actually use like a burglar alarm. And so when we're trying to collect these animals in the field, we have to sneak up on them and not cast a shadow over these eyes, or they'll clamp down. And so it's this really interesting system for studying biomineralization because we have these really cool teeth, which is what Rebecca's primarily interested in, but also shells, sclerites, and eyes with a mineralized lens, all in one convenient organismal package. And so Rebecca's been working to sequence the chitin genome using the same sort of approach. And so far Rebecca has collected Illumina data, several runs of Oxford nanopore data, and she's done optical mapping. And she has this incredibly high quality genome now. She's down to 84 scaffolds, which is near chromosome level assembly using this fairly affordable approach. And so using that, she's mined out the genes that appear to be involved in the biomineralization pathway of the teeth by sequencing radula transcriptomes from each of 4 different developmental regions of the radula. So the pre-mineralized, where iron begins to be added as iron is being transformed from amorphous to magnetite, and this senescent magnetite-rich layer. And she's doing differential gene expression analyses to identify the genes that seem to be involved in each of these phases, so secreting iron at all and converting it from amorphous iron to magnetite. And so she's trying to identify this biosynthetic pathway for this really cool hardened teeth in this cool animal. And so with that, thank you all very much for your attention and listening to me talk about my strange mollusks. And if there's time, I'd be happy to answer any questions. So this is using an Olympus scope, and so it has its own proprietary file format, which is a VSI, a virtual slide. But basically, you can export this as multiple TIFFs at different focal planes, and then they're just stitched all together with their own software. Microsoft has a product called Image Composite Editor, which allows you to stitch images like that together as well. Sorry, these are big. Yeah. So for example, like these animals are on the order of 1 centimeter. Yep. Yes? So good question. I can tell you that those that live in a sort of more burrowing habitat have more scale-like sclerites that are kind of adpressed to their body, whereas those that are epibenthic tend to be spinier. And so presumably the ancestral function was defense. The epibenthic ones are often very spiny. And then maybe those that secondarily move to a burrowing habitat have just kind of, they're along for the phylogenetic ride, and they've been made more, they've evolved to be more scale-like, so they're more vermiform. The caudofoveates do move peristaltically, yep. But there's been like zero functional morphological work done on that group. The problem is because many are deep sea, usually when they come up in a trawl, they're no longer behaving like a happy animal. However, off Fort Pierce, Florida, we can get myofaunal selenogasters that do come up. They're happy. They don't even know they're out of the sand. And we've started to do a little bit like looking at phototaxis and geotaxis. But when we put them back in the sand to observe those sort of things, we keep losing them. So that's the problem with working on myofauna, as some of you know. So we'd love to do more of that, though. So in Western Australia, if I don't get eaten by a great white shark, there are places where caudofoveates are like 50 per meter squared, and they're actually like 1 centimeter scale animals. So that's what we'd love to go and get animals for some more behavioral studies.
How are these animals responding to ocean acidification? And when you say deep sea, how deep are you talking about? Is it below, like outside?
Great question. So yes, there are some that are down to like 7,000 meters, and I forget what that magic number is, but there are some that are thought to be below that compensation zone. Most of the diversity is in the order of more like 1 kilometer. So deep, but not the deepest parts of the ocean.
And are they people done exposure experiments?
Oh, right. So really no. But I can tell you that when I've kept some that have these thin calcareous scales in the lab, like at Friday Harbor in a dish in the sea table, if I keep them overnight and the pH starts going down the next day, you can see that they're just looking kind of ragged, and we do SCM on them, and they're already beginning to demineralize. So even just a few worms in a dish with some bacteria growing begins to demineralize them. In museum collections, I often inherit these completely denuded animals where they've totally dissolved. And so if you don't rinse all the formalin out and actually buffer your ethanol, we buffer our ethanol with borax, often they decalcify. There's a whole family that's described as Gymnomeniidae because they're thought to be naked aplacophorans, but no, the museum ones were just all decalcified. Anything else? All right, thank you all very much.